Welcome everyone to a new stream. I got a good one for you today, and I think it's going to be a lot of fun. So we're gonna get into it pretty quickly. I know it's been a hot minute since the last stream, so I'm pretty excited to be back. And the topic of today, genetic engineering. I've had a lot of requests for making a video going into the basics of genetic engineering. And we'll talk about this in a minute, but I've done a bunch of videos in the past where we actually did some genetic engineering, but people sort of had issues where I had to glaze over some details, and I've always wanted to do a video where I could really get into the weeds, and I've written many scripts over the years. I've tried to put together a video that would kind of bundle it up into a nice package, and no matter how many times I did that, I realized that it's just not working. There's just too much information to try and convey in a single video. So instead, what we're going to do is we're going to break it up into little pieces. So got a lovely little presentation put together for you. And so one of the other reasons why I haven't done this particular video yet is that I wanted to make sure that when I do inevitably make the video, which is now, I was coming at it from a place of authority, and I'm not just sort of saying things without knowing that they're 100% correct, because genetic engineering is fundamentally extremely complicated, and there's a lot of little details that can trip up the average person. I know this because I've stumbled on them. So I wanted to make sure I had enough experience and enough, basically enough experience and enough, experimental work under my belt so that I was familiar with the most number of techniques and I could convey the information in the best possible manner. So we're going to be doing today's stream a little bit differently. And where in previous streams I would try and answer the questions as they came in, what we're going to be doing today is I'm going to just try and get through the whole presentation, and we're going to save all the questions for the end. There's a moderator in the chat, so all y'all behave, but also they'll be taking down questions, and I'm also going to be saving some questions. So if you have any questions while we're going through, just leave them there and I will get to them at the end of the presentation. This way I can kind of just flow through it without having to stop and lose my place. So, yeah, let's just get into it. Welcome everybody. I hope you're all having a fantastic day, and yeah, let's get into it. So, what are we going to be covering in this series? And I spent a lot of time. What just happened? Oh, there we go. I spent a lot of time really thinking through what would be the best things to convey and what would be the most important. But at the same time, I know the things that I think are important, but I don't necessarily know the things that you think are important. So if there's something that you want to learn about, then, leave me a comment and I'll be able to address that in future streams. And the nice thing with this is it's something I know really, really well. I'm already building these other presentations. So it's something that I'm hoping to be able to do more regularly. I know there haven't been a lot of videos, so this is kind of the new plan for being able to get some more content out. But so yeah, the things we're going to go through, building basic plasmids, because plasmids are sort of the core of genetic engineering. Even if you're doing stuff that isn't going to be a plasmid in the end, it's how you get the DNA where it's got to go. We're going to talk about DNA assembly strategies, because legitimately building DNA is one of the hardest parts of genetic engineering. Writing the code is trivial, but actually making it is far more difficult. We're going to go through a deep dive on promoters and different ways to control gene expression. We're going to talk about designing custom proteins. We're going to talk about gene localization, so getting expression of different proteins in specific places in specific organisms. We're going to talk about enzyme pathways. We're even going to get to genetic logic, which is some of the more cutting edge stuff, and it lets you do some really, really crazy stuff, and I think it'll be a lot of fun. And of course, whatever else you want to learn, leave me a comment. The presentation probably be in the range of 45 minutes to an hour. It's going to depend sort of how quickly I get through things. So today, though, we're really going to be talking mainly about what is genetic engineering, how is it different than biochemistry, synthetic biology, and genetics, and also sort of an overview of how the process works and how you go about it. Because the vast majority of the difficulty of genetic engineering is not the work itself. Like doing the lab work is essentially trivial. It's just moving small volumes of liquid around. The vast majority of the time and effort that goes into genetic engineering is spent planning your experiment and ordering the materials and collecting all the right materials. Because really, if you don't have your plan perfect, it won't work. And it's just that simple. Sound is a bit low. Is it? Is that a problem for everybody? I can turn up a bit, but anyway. So yeah, there's some comments about could I make a more compact video? And the plan is that once this series is finished, I'll have a pretty good basis for what is the information I need to kind of condense. And then I'm hoping to either cram it down into a couple of shorter, tighter videos, and also maybe a book. I've started writing a book on genetic engineering. It's going to take me a while, so don't like get your hopes up that it's coming out soon. But it is something where I feel like I'm in a unique position to write that book in a way that I don't think anyone else can. And there's a lot of things I want it to include, and there's a very specific form I want it to be, but it's also going to just take a while. So this is part of the reason why I'm doing these streams. It's so I can kind of get those thoughts together so I know what to include in the book. So that way there'll be eventually a handbook of this is how you do genetic engineering in the 21st century. Alrighty, so let's get into it. What is the difference between all these different things? Because you'll hear people use different words to describe genetic engineering, and while there's a lot of overlap in the choice of words and their meaning, generally there's some very subtle differences that people who are familiar with it would probably argue about, but also understand. So at its most basic, what is genetic engineering? And genetic engineering is modifying an organism to change their DNA or alter its properties. This could be anything. This could be any organism, doesn't matter. Whereas if somebody says that they're a biochemist or they do molecular biology, this could just mean that they're more in the domain of analyzing organisms rather than modifying them necessarily. So sometimes the terms molecular biology and genetic engineering are used interchangeably, but they don't necessarily mean the same thing. Synthetic biology is sort of the most modern of the lot. It's sort of an extension of genetic engineering, and I find it to be more specific. Like it has a specific goal in mind. Whereas, so like genetic engineering might be used in basic research. So let's say you want to modify a fly to see what a certain gene does. You could be engineering the fly, but it's not like you're trying to make the fly do something interesting. Whereas synthetic biology tends to be very industry focused and very output focused. So you're not trying to modify an organism just to see how it works. You're more trying to modify an organism because you want it to do a specific thing that you think is relevant, usually industrially. So this is something like modifying an E. coli or yeast to produce a specific protein or chemical that you want to be able to make in large quantities, rather than the more basic research stuff. Whereas genetics is more just the study of genes and how they interact and how they flow through populations, which frankly I find the most boring. But that's just me. I'm very execution focused. Like I want to see a thing do a thing. I don't really care how it spreads through a population, unless that's the thing I'm trying to do, So this is actually where I got tripped up when I went to university, because I took a program that was genetics. So it was a lot of like population genetics. It was seeing how genes flow through populations, like looking at deer populations, or how sickle cell anemia spreads through the population and the things that affect that trait, and dominant genes, recessive genes, blah, 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 blah. It's all fine. It's all interesting, and it lays the foundation for synthetic biology, but it's ultimately, I personally find it very boring. It's not what I'm interested in, but that's just me. There's still lots that can be done with it, and it's not to say that the research isn't valid. It absolutely is, and I wouldn't be able to do synthetic biology without this field of genetics. It's just not going to be the focus of these lectures or streams or whatever you want to call them, and it's also not, it's not my jam. It's not for me. But you know, this is just sort of a preference thing. Okay, let's talk about what are some GMOs, because there's some big distinctions on sort of what counts as a GMO or a genetically modified organism. Because, well, the problem is most people don't understand just how modified our world is. You have never eaten a food, well, okay, maybe not never, but you've almost never eaten a food that has not been distinctly modified by humans. Does that mean that we've gone in and purposely changed every gene? No, that's not what it means. But just through the act of breeding something, you will change its genetics. You will change thousands of genes at a time. So for people who are like, I don't like GMOs, it's like, why do you not like puppies? Puppies are wonderful. Cows are wonderful. Sheep is wonderful. Like every green vegetable that you eat, if it's cabbage, Brussels sprouts, kohlrabi, kale, broccoli, cauliflower, and a few others. This little image here does not actually cover the whole list. It's all the same plant. Like it's not just like, you know, they're similar species. It's the same species. They are the same plant, just modified by humans over thousands of years. So Brassica oleracea started as a it started as a mustard, basically. And that mustard was found by different populations of humans, and they just started breeding it for whatever they wanted it for, which is, you know, either terminal buds, lateral buds, stem, leaves. So, you know, like kale selected for leaves, whereas Brussels sprouts were selected for lateral buds. And just by selection and selection and selection and selection, you end up with something that is much more food-like. Because the things in nature are not good food. Not for humans, anyway. So this is a bit of a weird concept for people to understand. Like, an apple is just as man-made as a Pop-Tart. Like, there's no food in the grocery store that has not been touched by humans and made that way. Like, coconuts were domesticated. Mangoes were domesticated. Avocados should not exist. The only species that propagated avocados has been extinct for thousands of years. They only exist because humans like eating them. And we've bred them to be even larger monstrosities than they were when we found them. Corn. Like, corn is grass. Like, it's monstrous grass that has been bred over millennia to be what it is today. So it's kind of funny when people are like, I'm anti-GMO. I'm like, well, then don't eat. Like, don't eat and no puppies. Like, which seems a little bit harsh, but it is true. Like, and it's not hard. Like, humans have just casually genetically modified things without even realizing it. Like, dogs are not the wolves that they used to be. So, and, you know, people will be like, you know, breeding is different than GMOs. Is it? You're changing thousands of genes at a time. And then you look at something like nuclear gardening, where you take a plant. I mean, usually a plant. As far as I know, they haven't done this to animals, but I mean, don't quote me on that. But normally they take plants and they put them in a big ring, and they put a highly radioactive material in the middle, and they just irradiate the plant with enormous quantities of radiation to induce random mutations. Then they plant the seeds that come off of those plants, grow them, and if any of them have interesting properties that we like, we then backbreed that into a normal strain of that species. So, for example, the ruby red grapefruit. If you've ever had a grapefruit, you know, nice red fruit, it's not naturally like that. Like, it was made that way by exposing it to nuclear material, waiting for it to mutate, finding a plant that had an interesting property that we liked, which in this case was massive overproduction of anthocyanin, And then we bred that back into a stock of normal grapefruit so that we could adjust for things like taste and the stability of the gene. Because as you can imagine, after irradiating things, the genes aren't necessarily particularly stable. So you have to go through a whole bunch of generations of breeding afterwards to stabilize the line. This can also be the same even with modern GMOs. Like even if you just change one gene, you might have to then breed the plant a little bit to propagate both the trait that you've added and another trait that you're looking for that you know you can just breed in. So, you know, it's pretty neat. I mean, we've been doing this literally since, you know, we started agriculture. Like we've been doing agriculture as a species for, I think, like it's in the range of 6 to 10,000 years. There's some gray zone there. But basically as soon as you're doing agriculture, you're doing genetic modification. It just is the way that it is. But it's not purposeful. It's purposeful in that you're constantly selecting for bigger and bigger and bigger plants, or cows or fuzzier sheep or whatever. But you're not changing a specific gene on purpose. And that is where the line is between what I'm going to call modern GMOs and the original GMOs. So let's move on to the modern stuff. Now, this is where we get to the stuff that we're really going to focus on in these series, because this is really where the fun is. So there's a whole bunch of examples here, and we're going to kind of go through them pretty quickly. So the first one in the left is non-browning apples. So apples brown because of a single gene. Like there's a single gene that just makes them sensitive to oxygen, and it's highly pH sensitive. This is why if you take some lemon juice and like if you chop up some apples and then put them in a bowl with some lemon juice, they're much less likely to brown than if you just leave them out in the air. But that same gene that causes the browning, we can just get rid of. And so that's what they did. And they've now made non-browning apples. So you can cut them and leave them out and they'll sit there and they'll look perfect. But they just won't brown. The nutritional content is exactly the same. The flavor is exactly the same. The only difference is they just don't brown. So this is where modern genetic engineering really comes in, because you're able to make these very tight, controlled changes to a specific property of the plant or animal or whatever. Another one, which we've talked about before in a previous video, is golden rice. So one of the biggest problems with rice is that it produces essentially no vitamin A. And for people in impoverished countries, it can be very difficult to get foods other than rice. So people are basically subsisting on a nearly pure rice diet, which is not nutritious enough. So they end up being immensely deficient in vitamin A, and so they suffer unbelievably. So what a group of scientists did was they went, okay, well, if they're just going to eat rice, and we know that they can grow rice and afford rice, what if we just put the vitamin A inside the rice? And so that's what golden rice is. They took the genes for making beta-carotene, and they put it into rice. Now, we did the same thing, and we put it into yeast. And I'll show you a picture of that later. But you can take that same pathway and put it in whatever you want, and it makes this beautiful yellow-orange color from the beta-carotene that's produced. One that's a little bit newer, and I think is really where we're getting into the true synthetic biology space, is a company called Mufree. I think they've changed the name of the company at this point, or they got bought out by somebody else, so I don't know if this is the current name. But what they did was they did something very similar to what we did in a previous stream, which is they've modified yeast to produce milk proteins. Because really, what is milk? It's a collection of proteins and lipids and sugars in just the right ratio. But there's nothing to say that we necessarily need a cow to make those things. So if you make the proteins in yeast and you extract lipids out of plants that have the right consistency, and you extract sugars out of plants that have the right consistency, you can make something that looks and tastes and feels exactly and cooks exactly like milk without the cow. So this is where Mufree comes in. Another one that I think is probably the most common modern genetically modified material is actually insulin. Every vial of insulin that is sold on the market today comes from genetically modified bacteria or yeast. The vast majority of it is made in �E𝐿I. So it's really just an �E𝐿I that produces insulin. They extract it out of the �E𝐿I, they purify it, get it into a safe form, and then package it and sell it to whoever needs it if you've got type 1 or type 2 diabetes or any of the other times you might need insulin. And yeah, it's called Perfect Day now. Mufri is now called Perfect Day. Thank you for perfumed manatee. Anyway, back to insulin. So insulin, it's just a protein. It's a very small protein, and so it's very easy to make an �E𝐿I. And originally we had to harvest thousands upon thousands of tons of pig pancreases to make insulin, but this was obviously not cost efficient, nor was it particularly pig efficient, because the amount of pigs you needed to get a single vial of insulin was not a good trade-off, shall we say. But doing it with genetic engineering, where you just modify a bacteria and just have it do it, it's quick, it's easy, and it's efficient. And now the engineering is going into making better insulin. So these are forms of insulin that are better than what we could have ever extracted out of any natural organism. So I think it's kind of amazing. Like, there are people alive today. There are thousands of people alive today who could not be without this technology. And, this is one of the reasons why I get really frustrated when people are like, GMOs are bad. I'm like, well, why do you hate people who have diabetes? Like, they need to survive. Like, it's the only way they're going to be able to do it. It's a beautiful, wonderful technology. Like, chill. Another one, rennet. The way that we used to make cheese was we would basically take a calf's stomach and cut it into little pieces, and the proteins that are exuded by cow stomach include rennet. It's just this enzyme that causes milk to curdle. And so obviously this isn't particularly efficient because you need a lot of cow stomach, and it's kind of gross, and it doesn't store well, and it doesn't ship well because it's bits of cow stomach. So what they've done is the exact same thing as they've done with insulin, which is you just take the code for the protein, put it into a bacteria, grow the protein, extract it out, and then bottle it and sell it. And that's it. So essentially, every bit of cheese you've ever had is made with genetically modified bacteria. I think you might be starting to see a pattern. Every aspect of human existence is touched by genetically modified materials, and they're kind of the best. Okay, last one, soybeans. And not just soybeans, but all of the other plants that have been modified with the same mod, which is to make it glyphosate resistant. I mean, glyphosate, you are almost certainly have heard of this before. It's called Roundup. It's the chemical made by Monsanto originally. Glyphosate is a chemical that normally will kill most plants. And the thing with it is, because it kills most plants, it's actually a really useful herbicide. But how do you deal with the idea of, okay, well, I only want it to kill the plants that are not the ones I intend on eating? And the way that you do that is you just give glyphosate resistance to your plant of interest. In this case, soybeans, but again, it could be anything. Any of Monsanto's line of Roundup Ready crops have been modified with a single gene that make them resistant to glyphosate. And this means that you can apply glyphosate to the field and it will selectively kill whatever plants are not supposed to be growing there. Because of this technology, the amount of food we are able to produce as a species, and similar technologies like it, has dramatically increased. Genetic engineering is literally the only reason why there's as many humans on Earth as there are. Like, these technologies are what allow us to grow the food at the quantities and scale that is required to keep our society functioning. And so it's kind of amazing that literally a single gene and the right chemical is the difference between these two things. Now, of course, glyphosate, it has its problems, and there's all sorts of debate around this, even though it's been shown to be safe over and over and over and over and over again. But, some people don't like glyphosate, so that's fine. But it still lets us do things like another one, for example. If the problem isn't weeds that are interfering with your crops, maybe the problem is pests. So this is where the line of Bt crops come in. So Bt is a toxin that comes out of a species of bacteria, and it selectively targets insects, basically. So it only affects insects, and that's sort of the most important point. The toxin binds to the intestinal lining of various insect species, usually pests, and will basically perforate their intestinal lining, so it essentially causes them to leak to death, which is pretty gruesome, I realize. But previously, we used to just be growing the bacteria, like the natural bacteria that produces this stuff, extracting the protein, and then just spraying it on the crops. So the amount of Bt toxin that was on the crops was enormous. But what people did instead was they just took the protein and expressed it directly in the plants. So the actual level of Bt that is remaining in the crops by the end of the process is far lower. And you're not having to spray it on the fields, which means you're not having pesticides leak into the environment because they're contained inside the plant. So the only insects that are affected are the ones trying to eat the plant, which means it's only affecting pest insects instead of affecting the entire ecosystem. So it's literally better for the ecosystem when we do it this way. So I think it's kind of amazing when you look at it that way. Now, of course, things that are good for food are not necessarily the only things you can do with genetic engineering. For example, here's a selection of companies that are all doing stuff for purely cosmetic reasons. Now, let's go through them in order, because I think all of these are amazing, and this is really some of my absolute favorite GMOs, just because they are so beautiful. Okay, let's start on the left. So first up, we've got Light Bio, and their concept is very simple. They're making plants glow in the dark. And these aren't fluorescent, they're actually bioluminescent plants. So what they did was they took a pathway out of a bioluminescent species of fungus, and they transplanted it into plants. I believe they now have 4 or 5 different plants. I think they've got tobacco, which is what you see in the picture here. They've got petunias. I think they might be doing roses, and I think they have an English ivy. That's all bioluminescent. Like, this is the stuff of dreams. This is the stuff of, like, people watching Avatar going, I want a glowing forest. And then somebody went, yeah, I do want a glowing forest. I'm going to make one. And then they did. So these plants, I don't think are available for sale just yet. I think they're currently in talks with the EPA and a few others about just making sure that it's safe to release them. Because I believe, and I don't know their technology like intimately, but I would assume that they're having to do this in a way where the gene can't escape from the host species. Because, I mean, as wonderful as it would be if all of our forests started glowing, that would probably freak a whole bunch of people out and screw with some ecosystems. So they're making sure that the plants are highly contained in the sense that, like, they can be safely grown in like a home environment without worrying that the gene would get out and get into another species and, like, you know, your oak tree starts glowing. I still don't see this as a downside. I see this as an absolute win. But, you know, government agencies take these things very seriously. So, yeah. But anyway, the second those things are available, you can bet that I'm going to be one of the first people to buy one. And if they're watching and they want to, you know, sponsor the channel, you know, send me an email. That'd be fun. Alrighty, moving on, we have glowfish. So these are actually things you can buy right now. And they're various species of fish that have been genetically modified with fluorescent proteins taken usually out of coral. So corals produce these special proteins called fluorescent proteins that, I mean, it's literally what it does on the tin. It says on the tin, you know, like it's a protein, it's fluorescent, they're highly colorful. And so they've taken albino species of fish and they've just expressed these proteins in usually the skin and muscle tissue of the fish. And as you can see, it makes for beautiful, beautiful fluorescent fish. And they're gorgeous. Like, they're really cool to have as an aquarium addition. And yeah, I mean, there's not much more to say about them. They're just really pretty fish. But this is the sort of thing that you can do when you kind of do genetic engineering really carefully. And last but not least, certainly not least, we have 𝐎𝐿𝐼𝐿. So what these guys are doing, and for full disclosure, I am actually working with them. I do need to say that. So what 𝐎𝐿𝐼𝐿 is doing is they're taking the systems from bioluminescent species. So, you know, things like fireflies or, you know, glowing bacteria or mushrooms or whatever. And rather than like trying to get an organism to glow, they're producing the components, so the glowing enzyme and the chemical that it needs to react with to cause the glow, and they're producing it at enormous scale so that they can actually make a glow stick or glowing bioluminescent products that aren't alive. Like, it's literally a powder that you add to water and it just starts glowing like magic. I've been playing with this stuff for the last probably 6 months, and I kid you not, it's magic every single time I use it. It's absolutely gorgeous, and it works. It's crazy watching it work. Like, it's one of those things where if you were to ask like any kind of bio nerd like myself who does genetic engineering, like, if you could do one thing, like, what is it? Yeah, bioluminescence is almost always top of the list, and they've done it, and it works, and it's awesome. So yeah, highly recommend checking them out. They're really dope. They're also just really dope people. So yeah, check them out. Very cool. And yeah, people are asking, you know, could it be not be made chemically? And no, you can't make the enzyme that does the glow chemically. You can make because it's an enzyme and a chemical together. You can make the chemical like in a normal chemistry lab, but without the enzyme, it's useless. It doesn't do anything. So they grow the enzyme and they produce the chemical, and then they package it together and sell it as like a glow stick or for whatever bioluminescent product you can dream of, essentially. So yeah, they're super dope. All right, so these are some of the ones that we've done in past streams and past videos, just to kind of give you an idea of, you know, if there were no rules and we could just do whatever, like, what can you really do with genetic engineering? And the answer is basically whatever you can dream of and afford. And so, you know, we've done things like coding a yeast that will produce milk proteins and egg proteins. We wrote up the code to knock out caffeine production in coffee beans, which most people thought was pretty cursed. And yeah, that's not even the least cursed genetic thing I've ever come up with. But it was fun, and it let us really explore some very interesting methods for throttling the production of proteins in an organism. We did a penicillin producing yeast. We did spicy tomatoes. And these were just the streams where we actually got into the nitty-gritty of designing the code, doing the research, figuring out what gene needs to be modified, modifying, and then designing the code that would do that modification. With the exception of the milk and egg yeast, I haven't actually made any of these, but the milk and egg yeast, I did actually have the DNA made, and I grew it, and it kind of works. It had a whole bunch of problems, which is the reason why I haven't made a video about it yet. It sort of needs to be redesigned because the original design had some issues. Primarily, yeast are a pain in the ass and very, very finicky. So they did produce the protein, but it was at such a low level that it wasn't like useful. So it's not like, because the idea behind that one is I really wanted to grow a cheese omelet, and yeah, the level is just too low. The amount of yeast that I would have had to grow just to make like a tiny, like hamster-sized omelet would be just unbelievable. So it wasn't worth the effort. So it needs to go through a redesign process. These are some of the videos that we did on where we actually did the genetic engineering. Like we did the experiment. And we'll just go through them really quick. There's more than this on the channel, but these are just some like representative examples. And it's the same with the streams as well, right? Like there's a lot more streams than you see here. This was just sort of a representative sampler. So in terms of the actual videos, we did growing synthetic spider silk in yeast. And this one is kind of the one that's sort of the closest to my heart because I've been working on it the most. And I mean, it's also the reason why there haven't been any videos. It's what I do literally every single day. Like literally, I wake up, I work on spider silk, I go to bed, I'm still working on spider silk, then I wake up the next day and I'm still working on spider silk. It's all I do every single day, and you know, I'll talk about it when I can talk about it, but I can't talk about it yet. The next one we did was the beta-carotene yeast. So the golden rice that I talked about a minute ago, like I said, we took that exact same pathway and we expressed it in yeast, and you can see it gives it this beautiful yellow color of beta-carotene. And then, of course, I also have a food channel, so we baked some bread with it. And, I mean, it made some pretty good bread, I'll be honest. Probably the one that got me the most notoriety was this one, which is fixing my own lactose intolerance with a homebrew virus. That one was crazy. But yeah, lactose intolerance is caused by a single gene turning off. So, I'm a bit of a nutcase. So thought, okay, well, my life is basically pain because they put lactose in everything, and it would like sneak up on me. Like I'd eat something and not know that there's lactose in there, and then I'm like hiding in the bathroom for the next 3 days. So at that point, the cost benefit relationship in my head was sufficient that I was like, okay, yeah, I'll try and fix this. I don't recommend this, by the way. Don't copy me. It's a bad plan. I'm an idiot. Don't necessarily follow in my footsteps on that one. But it did work. The virus that we grew did end up delivering the gene to my intestinal cells, and I could eat lactose for about 1.5 years before it started to wear off. These days, it's definitely worn off, but I can take a lot less lactate than I needed before, so that's fun. But it did work. But I'm not going to be rushing to redo that one because the cost benefit relationship was like it made sense at the time. But at this point, my lactose intolerance is much more tolerable. So it's not that I feel the need to take it because my quality of life is low. So it's much more manageable. So I'm not going to risk it. Because I mean, genetically modifying yourself comes with a huge amount of risk that I really don't recommend. Like I did it because I was in a very particular situation and I had the experience and it made sense, but like, don't do it. It's a bad plan. And yeah, then finally, the last one's very simple. We just took that same fluorescent protein that they put in the glowfish and I just put it into some 𝐒. And it just makes them glow. It's very fun. It's easy. And it's one of the most basic things that you can do in genetic engineering. And the next stream where we actually get into how to make DNA, like how to code it, how to write it, like what are the pieces, what's going on. We're going to probably start with making a fluorescent protein plasmid because it's simple, it's easy, and it's obvious when it works. Like the problem with genetic engineering a lot of the time is you're doing stuff like you're modifying a protein that doesn't have any color or other indication that the thing worked. So you have to do a whole bunch of testing to figure out if the gene mod that you made actually is working properly. Whereas the fluorescent protein is really obvious because like you go from a beige bacteria to one that's like neon hot pink and you're like, okay, yeah, it's probably working. But anything more than that gets really complicated and the testing gets really hard. So it becomes more difficult and there's just a lot more to convey. So we'll start with the simple case and move on from there. Okay, so now we're going to get into the actual, like, how does this work, right? Like we've talked about all of these different things that people have done with genetic engineering, but how does it actually work? And the answer is it's not that complicated. So like when we think of a computer, right, like to be able to write code for a computer, like there's different levels of abstraction depending on what you're trying to do. So, you'll start at like the bare bottom of, physics and then like how transistors work, gates, combining gates to make different architecture, instruction sets. Then you get to like assembly machine code. Then you get to your like programming language, your algorithms, your applications, right? Like it's that progressively more complicated, but also more abstract. So like if you're writing Python, you don't need to know what the bare metal is doing necessarily, right? Like you can just write Python code and have no idea how a computer actually works. I don't recommend it. You're not going to be a very good programmer, but it is possible. Genetic engineering is exactly the same. So with genetic engineering, it's genetic abstraction. So you start with physics, and then you've got chemistry, and then from there, it just kind of builds up. So you've got DNA letters, then you've got DNA codons, so, sets of 3 letters. Then you have genetic components. So this is going to be things like promoters, operators, terminators, this sort of thing. Then above that, you have the actual output of these genetic components, so generally proteins. From proteins, you go to collections of proteins. So these are what we call genetic devices. So this could be a collection of proteins that do a specific thing. And but it can also be a very small subset of things. So you could take a protein. So for example, there's a set of 3 proteins that they found in, it's some sort of bacteria. I don't remember off the top of my head, but it controls the circadian rhythm of the bacteria. And, it's just these 3 proteins that work in a cycle where, one works on the next or works on the next, and one of them is photosensitive. So you can control the circadian rhythm just by applying light to these 3 proteins, and they'll change states and then affect things down the line. So that's a genetic device. It's just a small collection of stuff that does a thing. Beyond that, then you're getting into molecular networks. These can get enormous. So this is where you actually want to go from, I'm going to make one protein, to I want to make a chemical. Making a chemical with genetic engineering is infinitely more difficult than making a single protein, because you generally need anywhere from 2 to 15 different enzymes all working together in exactly the right proportions and at exactly the right rate to get it to do the chemistry that you want it to do. And then you've got to go in and you've got to tweak each individual protein and do all kinds of little mods to it. It gets really complicated really quickly. But there's also molecular networks just in every organism. Like at the end of the day, you're basically just a cascade of chemical reactions happening that are so unbelievably complicated that we can have this conversation, right? Yeah, that's a molecular network. Above that, then you've got your organism level. So this is where you're choosing which organism you want to work with, be it E. coli, yeast, plants, whatever. Then above that, you've got the actual culturing of the organism. So now you're growing whatever it is that you want to be growing, and you're controlling the molecular networks and proteins and stuff that are going into that grow. And then from there, you kind of have the step which most people don't tend to interact with, but it's sort of where I'm spending most of my time, and that's large scale culture. So this is now you've modified a thing to do something, and you want a lot of it. So this is like growing tanks of something that are 30,000 liters per tank of E. coli or whatever to do the thing that you're trying to do. But yeah, it's just these levels of abstraction, and the deeper down you go, the more finicky it is and the more detail you need to know. But where it's different, where genetics is different compared to a computer, with a computer, you can write Python code and not have any idea how transistors work. With genetics, you do not have that luxury. You need to know every single level. You need to know how the organism works. You need to know how your pathway works. You need to know how your devices work. What's the protein look like? How's it work? What's important about it? How are your genetics? What is your code down to the letter? You need to know the chemistry involved. You need to know the physics involved, because sometimes all of it's at play at the same time. So it's very, very complicated. There's a lot of information you need to know. So to be a good genetic engineer, it's really a game of learning as much as you physically can. You can start off pretty easily not knowing all that much, and you can do basic stuff, but to do it well is a game of knowledge. Every technique you know, every fact you know, every weird organism that you've heard of, every weird protein that you've heard of, is a new tool in your toolbox that lets you do genetic engineering better. And without that, you're basically screwed. And it's just not going to work. Like I say, planning and understanding and having a good story in your head of what is going on at every level on this level of on the the scale of abstraction is important. You have to know what is going on. Otherwise, things happen and you don't know how to test for them. You don't know how to do, well, basically anything. So it's super, super important. But at the end of the day, this is essentially the steps on how to do genetic engineering. And it doesn't seem like a long list, but believe me, it will it will break you if you're not prepared for it and if you're not ready. So we're going to go through this step by step and look at how to succeed at each step or sort of give some insight on how to succeed at every step. And breaking it down into little pieces is really going to be your best bet to have any kind of success. So step number 1, pick a thing. And legitimately, this is actually something that people have a lot of problems with, because most people get very excited and then have no idea what they actually want to do. So for me, like, silk has always been my thing. Like, where most people are like, I want to be Spider-Man when I grow up. I'm like, I'm going to be Spider-Man when I grow up. You know what I mean? Like, I knew what I wanted and I did it, and I'm still doing it. I do it every day. Whereas, some people are like, I don't know where to start. Like, find a thing you're passionate about, because, and I think this is probably the best piece of advice I'll be able to give you. If you're not really, really, really excited about the thing you're going to be trying to do, when it gets hard, you're going to give up. Biology is hard. You need to be in it to win it, because it's not going to work most of the time. The overwhelming majority of what you do in a lab is fail. Like, your things break, nothing works, you don't know why, you feel like you're an idiot, like, you feel like you're going crazy, like, you go through these cycles of like, am I an idiot? Do I know what I'm doing? And then it works, and you're like, I am a god. And, it's very hard to, keep a sane mind as you're doing this. But, picking a thing that you really care about is sort of the easiest way to do that. So first things first, you got to pick an organism. And this is really going to dictate heavily what you can do. So, if you want to treat a disease, you're going to be looking at animals. Whereas if you want to grow a new crop, you're going to be looking at plants. Whereas if you want to make a protein or a chemical, you're probably going to be looking at something like an algae or some other microbe. But there's a lot of things that you can do once you pick something. So just to quickly go through a few of these, a lot of these are just stock photos, but the one that I really want to highlight is the red plant in the middle. So this image was taken from a friend of mine, Sebastian. His handle is a tiny green cell on most platforms. It's fairly easy to find him. His name's Sebastian 𝐎𝐫𝖢𝐠𝐢𝐳. Really great guy. But this plant is, I believe it's a tobacco that he modified. And it's producing a pathway taken out of beets. So, beets are red. They produce a red chemical. That's why they look like that. And so he took this, I think it's 3 proteins that work together to make that chemical, and he put it into this plant. And as you can see, the effect is overwhelming. It is very red. The particular construct that he used was not tissue specific, so it just made the whole plant red, like deep red. But it's amazing. Like, it's just such a simple thing, but it's awesome. Like, these little changes can make such a profound difference to what a plant looks like and how we interact with it. But I also, really, the reason I wanted to highlight this is because just look at those roots. They're so cool. Like, they just look so alien and so weird. I love it. The other ones are not that interesting. The yeast one here is just various flavors of, beta-carotene expression and, like, knocking out different parts of the pathway. So you end up with different parts of the chemical has had different changes made to it, which is why you get the different colors. On the far right, we've got some 𝐒. coli that are modified with a fluorescent protein. On the left, we have some animals that have been modified with that same fluorescent protein. And you can see it makes them glow bright green. Most of the time when you make a fluorescent animal, though, you're not doing it because you just want to make, like, a fluorescent animal. You're doing it because fundamentally you're testing something else. And like I said, if you can't see what you're doing, it means all testing is very hard. So having a fluorescent marker to tell you which cells are modified just makes everything so much easier. Like, when you're not in the final stages of, like, this is now a product, and you just want to be doing the testing, it just makes, like, sorting through your stuff easier. Okay, so now you've picked an organism. Now you got to pick a thing. So it could be a protein, it could be insulin, it could be silk, it could be a fluorescent protein, it could be rennet, or maybe you want to make a chemical. So you've got, beta-carotene, you've got maybe drugs or, flavor compounds or whatever it is that you want to make. If there's an industry that uses it, there's an argument for making it using synthetic biology. But I mean, ultimately, this is going to be a very personal choice. It's going to be whatever it is that you're interested in. And I can't make that decision for you. But ultimately, you just got to pick something. And once you've picked it, then you go through the process of figuring out, okay, is this even doable? But step one, just pick a thing. So the next part is break the problem into parts. So, you know, this is probably the most important step of the whole thing. If you want to do something very weird, it's going to be very hard because there's just not a lot of literature necessarily on doing that thing. So it means you're going to have to tread a lot of that ground for the first time. Anything that is like that is going to hurt. Like, it's going to take a lot of time and a lot of money to get it to work. Whereas if you're trying to replicate something that's already been done, you're probably going to have an easier time of it. So, for example, like making silk is not like a new thing. Like, it's been done many times in the literature, which is part of the reason I was able to, you know, do it at all. You know, I have that literature backing to draw from to help solve problems as they come up. Whereas, let's say you wanted to make cat girls. That's going to be really hard. There's no research on this. Zip, diddly, nada, squat. So you're essentially shooting in the dark, which means all of the research has to be done from scratch. It's going to take you a long time, and it's going to be very hard and very expensive. The next thing that you need to keep in mind is how many genes are required to do your thing. So if you're making a protein, the answer is usually 1. Whereas if you're making a chemical, the answer could be anywhere from like 2 to 15. Every gene you need to include in your construct makes it harder and makes it less likely to succeed because there's more moving pieces that all need to work in concert, and it's just so much harder. Like if you're trying to make beta carotene, I believe it's 4 different enzymes, and like that's not bad, but that's already a lot. So going through the process of trying to make it at high yield becomes very difficult because you're like, okay, well, which of the 4 do I need to modify? Which ones need to be in the right proportions? You're trying to balance the levels of each enzyme to get it to do it at exactly the right level. You know, one of the things that happens in beta carotene production is that one of the enzymes is limiting and it's just slow. Like compared to like some of the enzymes are very quick and they do their step very quickly, whereas some of them are not. And so you'll end up with a thing that looks very red because, say, the step that goes from lycopene to beta carotene is slow. So you're going to have to go, okay, well, if that's the slow step, then I need more of that enzyme to speed that step up to increase my production or whatever it is you're trying to do. The next thing you need to think of is, what do you actually want to do with your thing if it works? Because, and this is, I feel, where a lot of people get stuck, where they get so focused on just making it work that they miss the "and then what" question. And, you know, the answer doesn't necessarily need to be anything. Like, it could just be, okay, you've done something cool, publish it. Like, that's it. But, I mean, that's fine. You know, that's cool. That's helpful. But sometimes the thing that you've spent all this time and money and effort doing is worth a lot of money. So, you know, you're going to want to go, okay, well, can I industrialize this process? And if that's the route you plan on going, as you're doing the initial work to make your thing work, you have to be taking into account that things that you do at lab scale probably aren't going to work at industrial scale. So you have to be keeping those things in mind and accounting for them. Otherwise, you're going to get to the end, have a thing that basically works, and then go, okay, now I got to figure out how to grow it at scale, only to figure out that one of your steps just does not scale, and then you're screwed. So it's sort of a very important thing to keep in mind. The other thing to keep in mind is it medical, food, or an animal? Often, any of those things, and in fact, almost always, those things come with enormous amounts of legislation that you have to contend with if you want to commercialize your thing. This is why you can't buy the glowing plants yet. Like, there are laws in place that prevent you from doing that. And on the topic of laws in place that prevent you from doing the thing, before you try and do any genetic engineering, you need to be aware of what the regulatory environment that you're in is like. For example, if you're in the EU, doing most genetic engineering is illegal without a license. Whereas if you're in America, you can do almost whatever you want, like within a range. In Canada, the only things that are legislated and controlled are if you're making anything that works with something infectious, something that makes a mammalian toxin, like something that can like is toxic to mammals, or if it's infectious, I said already, but like so viruses, infect like toxins, and also if you're going to work with anything that has a spine. So if it's a vertebrate, you're not allowed to work with it without a license. So no working with mice unless you have a license. But like anything else is sort of fair game and you can do whatever you want. But you need to know these things before you do it, because the last thing you need is to get all the way through your project just to have, big daddy government show up and go, surprise, you get prison. That's no good. Nobody wants that. So you have to be aware of what you're doing. These things are dangerous if they're not done properly. You need to be aware of what you're doing, and you need to be very careful, both legally and physically. And then the last thing is also where people get tripped up is, what's this going to cost? Biology is expensive. Genetic engineering is unbelievably expensive. And I don't want to say that to like scare you. It's just the reality. Every reagent costs an enormous quantity of money. Like a bottle of 𝐎𝐫𝐢𝐠𝐢𝐳 resin can be like $1,300 for 100 milliliters of the stuff. And you'll go through that resin really quickly if you're doing anything at any reasonable scale. So you have to be aware of this going in. If you think that you're going to be able to like get through with garbage and whatever, it's not going to work. Like I know a few people who, like Sebastian, for example, like he takes a lot of pride in being able to do genetic engineering on a really low budget, but he's still spent a lot of money over the years building up his collection of reagents and materials. And he has to be very, very careful with what he actually does, because he can't afford necessarily to spend tens of thousands of dollars on a project should something go wrong. So you have to take even more planning to make sure that you're not just pissing away money on stuff that's not going to work. So it's a lot of, it's a lot of the problem. Like genetic engineering is really, really, really expensive. You need to know this going in. The cute little kits that you get from like 𝐎𝐵𝐼𝐻 or the fuckheads at the �H𝐵𝐲𝐷, like they don't actually acquaint you with what this stuff costs. It is unbelievably expensive. Getting DNA printed, very expensive. Buying reagents to put DNA together, very expensive. Hundreds and hundreds and hundreds of dollars. You need to be aware of this. But if you plan accordingly, it's fine. Like this is why a lot of the genetic engineering is done by academics. It's not because they're the only people who can do it. It's because they're the only people who can afford to do it. And that's really it. But the technologies are getting cheaper. The reagents are getting cheaper. And if you plan your thing carefully, or if you build your lab supplies slowly and build up to it, you can do a lot with very little. So it's a bit of a balancing act. Like you've got to be able to know when is the right time to just spend the money and get the proper thing, and when is the time to spend the time and build or try and cobble something together for cheap. So it's always this balance of time versus cost versus available resources. And it's just you have to be aware of this, or you're just not going to get anywhere. All right, moving on. Step 3, research. This is a really important step. If you if you're going to do genetic engineering, you have to have a plan. And the only way that you're going to have a plan, and the only way that you're going to know what's going on, is from research. This is why I say that if you have to do all the basic research yourself, it's just going to take you forever, because you have to do so many more experiments than if you're trying to do something that's at least a little bit established in the literature. If you're the first one to do something, it's very hard, but you have the novel factor to it. So, if you're the first one to publish it, you get all the credit, but it also comes with enormous challenge. Like, this is why people get handed Nobel Prizes, because they're the first to figure something out. And it took a lot of effort to do so, usually. So, it's an important thing. Now, in terms of where to go to find this research, these are sort of the ones that I like to use. NCBI is a phenomenal repository of all sorts of papers. ResearchGate, there's all kinds of papers. ResearchGate is also really interesting because it's kind of like a Stack Overflow for genetics. So a lot of the times, some of the basic techniques that everybody does, people will run into problems with it. So if I get stuck on a protocol that just isn't working for some reason, a lot of the times I'll go to ResearchGate and see if I can find somebody who's had the same problem. And then it might just have the answer. It's like, oh, okay, you just need to, boil the thing for an extra 2 minutes, or you have to add this extra reagent you weren't expecting, or maybe try this alternative protocol that works better, or whatever the case may be. Another really fantastic resource is iGEM. So iGEM is the genetic engineering competition that happens every year, and all the teams are required to submit all of their code and do a big write-up of everything that they're doing. And the iGEM teams are always so frickin' clever. Like, people just come up with the coolest stuff. And there's just such a wealth of information available that it can be really helpful to just go through what other people have done. And just, it's great for giving you ideas for like, ooh, hey, I could take that idea and then tweak it and do something else with it. Or, maybe I can use a piece of code that they developed and use it in a new way or whatever. So it's also a really great repository of like genetic pieces that you can use. A far less used and known journal is called Jove. And this one is really interesting because their whole thing is video. Like, there is often a write-up, but the main thing that they do is video protocols. Now, Jove is very expensive, so I don't recommend necessarily getting a subscription, but a lot of the time you can find the videos for free on places like NCBI, or, sometimes you can like torrent them or whatever. But the nice thing with Jove is you can actually watch somebody do the protocol. When you just read a protocol in a paper, people leave out details constantly. Sometimes they do this on purpose, but a lot of the times they do it because they just don't think it's an important detail, even though it absolutely is. So being able to watch somebody do the thing can be very, very helpful, because then you can actually catch little details that you might not have known were important as you were doing the protocol. And, it's just really helpful. There's also just weird stuff on there that, like, it's just neat seeing how people do things. And then, of course, we've got to talk about, the most important one of the list, and that's Sci-Hub. You see, scientific publishing was invented by a capitalist shithead in the 50s, and they decided that all that research that was being paid for by people's taxes, well, nobody should be able to get that unless they pay the publisher money, even though the publisher does nothing other than host the content. And these days, that means it's being put on a website. So, yeah, Elsevier can, I believe the technical term is eat a bag of dicks. So, highly recommend Sci-Hub. It lets you get every paper on Earth that's ever been written for free. So, yeah, highly recommend it. It's a really, really amazing resource, and I could not do my job without Sci-Hub. In fact, if you were to ask, essentially, any scientist, they would tell you they cannot do their job without Sci-Hub. If you're very lucky and you're like in a university, or you've got access to a university library, they may have subscriptions to some of the journals. So you might be able to get some articles. But like, publishers charge $170 per read. Like, just to read one journal paper, they'll charge you $150, which is ridiculous. And or like, just a subscription to a journal could be like $3,000 or $4,000 a year. It's absurd. Again, also they can paywall stuff that was paid for with your taxes. So yeah, I recommend Sci-Hub. It's technically illegal, but it's also forced all of the publishers to start doing open access. Because Sci-Hub exists, more papers are now available as open access than ever before, because it's literally like carrot or stick. You can either open up your stuff, or Sci-Hub will steal it, and then everybody can use it, and you don't get paid. So it's kind of amazing. Highly recommend them. It's also probably the most visited site. Like, it has literally all of human knowledge for free. Like, any paper on anything you could ever dream of, available on Sci-Hub. There is a time delay, so the newest papers tend to not be available right away, and that's one of the downsides. So if that happens, it's really good to hop on to, try and find various groups. Like, there's a few Facebook groups, and there's a few like different forums and stuff where scientists like to hang out. And a lot of the time, you can either email the author of a paper if you want it, and they're allowed to send you a copy of the paper for free. Like, the publisher doesn't get like exclusive rights. Like, if an author wants to just send you the thing, that's fine. Or a lot of the time these days, you can find the preprint version of the paper on bioarchive, which is also another great resource, which I regret not putting on this presentation, but I should have. Yeah. So yeah, so you can either contact the author or use Sci-Hub. or try and find a friend who's got access to like a university library and can just get more recent papers. But generally anything up to like 2021 Sci-Hub usually has. It's literally like the last year is where it tends to be a gray zone where you don't know if they're going to have it or not. Because generally what happens is they'll post a bunch of papers, then they get sued again, so they stop posting papers for a little bit. Then they either win the lawsuit or they just stop caring, and then they publish a whole bunch more papers. Usually the latter, because this Sci-Hub is run by a fantastic lady in I want to say it was Kazakhstan. But either way, it's basically impossible to take down now, and thankfully for that, because it's legitimately it is moving science forward more than just about anything else on Earth. So, you know. Alrighty, designing the DNA. This is the actual, like, I keep saying that this is the hard part. It's all the hard part. It's all hard. Like, there's no easy part. It's all hard. But anyway, designing the DNA. Now, there's a bunch of different ways to do this. We're not going to get too much into the weeds of how you actually go about this today. That's what the next episodes are going to be. And also, if you want to see actually designing some DNA, then any of the Whose Gene Is It Anyway streams, which I've linked below, there's, you know, you can see me doing it there. You can see a little bit of it in some of the other videos that I mentioned. But we're going to, in this series, we're going to get into the weeds of, like, designing DNA. But the important things to know are these are some of the tools. These are definitely not all of them. This is just the ones I'm personally familiar with that let you edit DNA on a computer. Because again, planning is everything, and these are the way that you actually plan your stuff. So, for example, this is what benchling looks like, where you have, you know, your code on the left, a view of what the plasmid looks like on the right. You can see all of the different like genetic pieces. So, you know, your promoters, your operators, your different proteins, your resistance markers, origins of replication, whatever the case may be. You can see how it's all laid out, and then you can go in and start changing things and designing your experiment to first modify the DNA and create the DNA the way that you want it, then put it into the organism that you want and have it do whatever it is your thing is. But like I say, I'm not gonna dwell on this too much because we're gonna get into it more in the next episodes. But this is what it looks like. Like, you literally change individual letters as if it was binary, basically. Like, it's this really weird mix of, like, abstraction and changing individual letters. Because I don't know what this, like, when you look at a protein, you don't necessarily know what every letter does, right? Like, the protein is the protein. Once you've studied it enough, you might know, okay, if I just make this change or this change, I might change something. But generally, you just know, like, this protein is coded for by this sequence. But then you could, like, go in and go, okay, I'm going to keep all the amino acids the same, but I need to change this DNA letter because of how I intend on putting the thing together. And if this letter's here, it's going to interfere with that process, or whatever the case may be. Like I say, we'll get into that more in future episodes, but I just wanted to show you what this looks like for anyone who hasn't seen this before. But yeah, it's fancy rings. Okay, step 5. Once you've actually designed the DNA, you order it. And then you've got to put it into your host organism, whatever that may be. And that step can be very difficult depending on what you're doing, or it can be very easy. It depends on what your thing is. In terms of ordering, there's a whole bunch of different companies, and it depends what you're ordering and what you want. The one that I personally use the most these days is GeneScript. This is not sponsored. I wish they would sponsor me. That would be awesome. I love their service. But this is not sponsored. Their ability to make DNA has been consistently impressive, and I really like their online like ordering system. But now there's a really big but with all of this, which is that a lot of these companies that are capable of printing DNA are not going to ship to a residential address. And this is going to be a recurring thing that you'll run into with essentially every bit of bio that you're going to want to do. Biology companies do not want to ship to residential addresses, and they don't want to ship to individuals. What they do want to ship to are commercial addresses and companies. The thing is, though, they don't actually check, like, what kind of company you are, nor do they care at all. Like, all you have to do to be able to order from one of these companies is just start a company, which is usually like a bit of paperwork and a couple of fees of like $20. And you can start like an LLC or a sole proprietorship or whatever the case may be. And then you have a company. And then if you rent a space that's in a commercial lot, you now have a commercial space. Like, you don't need a huge space for biology necessarily. Like, bio can be very condensed. And, academic institutions, like they'll also ship, like, especially they'll ship to academic institutions. So if you have any kind of ability to, have a mailing address at any of those kinds of places, then you can use that. But really, the best thing that I can recommend is that if you're really serious about doing biology, and like you really, really are serious about it, rent a little space, start a little company, and then you can order whatever you want. And there's no issues or problems or whatever. But yeah, like I say, there's other companies. I haven't used IDT or GeneWiz. I've heard they're good, but I have no experience with them personally. Thermo Fisher is one of the largest suppliers of biomaterials generally. But the main thing I use them for is printing oligos, which are short little single-stranded pieces of DNA used as primers for PCR and similar techniques. So I like Thermo. They ship really fast, and they're good for the oligos. Their prices are okay. They're not the best, but their quality is fantastic. And like I've never once wondered, did they misprint my thing? Whereas there was another company that I used to use called Alpha DNA, which I became very suspicious that they were misprinting the primers I was ordering, so I stopped using them. So yeah, if I've got to give one recommendation, I would say GeneScript. Again, not sponsored. I just like their service. All right. Now, the actual putting of the DNA into things, like I say, it can be very difficult depending on what you're doing. But if you're doing microbes, it's not normally that bad if it's a known microbe. So E. coli are one of the most studied organisms on Earth by far. Like, we know basically what all the genes do, and the number of different methods for getting DNA into E. coli are vast and easy and fast. They're vast and fast. And it just makes them really easy to work with. Like, so, in the video on the left, this bacteria is a nightmare, I kind of actually went overboard. Now that I have proper equipment and, things like a -80 freezer, it's actually even easier to get DNA into bacteria. Like, I don't even think about it anymore. Like, these days, I just pull a tube out of my -80, throw some DNA in it, heat it up, cool it down, plate it, we're done. Like, it's that easy. Yeast tend to be more finicky. The recipe for the solution that you use for putting the DNA into them is more complicated, has more ingredients, it's more expensive, and the amount of DNA that you need is unbelievably huge. So, for example, E. coli, you can get away with like 10 nanograms, 50 nanograms of DNA, whereas yeast, you're looking at a microgram of DNA at a minimum. So anywhere from 50 to 100 times more DNA as the starting place just to get any kind of transformation efficiency. Like, the amount of DNA is ridiculous. And so this just makes everything more expensive. DNA is not expensive per se, but if you need a microgram to do a thing, it means you've got to prepare a lot more DNA. Whereas with E. coli, you basically fart DNA at them and they sort of just take it and do it. So yeah, this is the thing. But anything beyond these get very hard. If you're going to try and modify plants, it's a whole thing. Like, it can be very easy, but it can also be very challenging because there's a lot of other steps that aren't the actual modification step that are very hard that you have to do before you can do the modification. For things like animals, it's a little weird. Like, if you're doing animal cells in a dish, it's very easy, but if you're doing like a whole animal, it's much more difficult. Because then you've got to work with embryos and all that kind of stuff, and it's just a nightmare. You're looking at micro injections or any of the other ways to do it. It's really hard. But if you're doing it with a dish, you basically just throw the DNA in there with the right reagent and it just does it. So it's a bit of a mixed bag. The other problem with animals though is if you're doing it in a dish, the DNA is not stable. It doesn't stick around. So, the mod will only last for, a couple of generations, and then it just loses it, basically. There are ways to make stable lines, but it generally people do. Sorry, I have hiccups. Generally people do a temporary thing, and then by the time they're ready to throw the culture out, the DNA is already fading, but you've already got your data, so it's fine. But yeah, so that's actually doing the thing. Like I say, these videos, if you want to see me do this and put some DNA into things, there's some very decent protocols in here. And I'm hoping that I'll be able to do more in the future and show more organisms and improved protocols for all of this. Oop, too far. Okay, step number 6. So let's say you have done the thing. You've designed the DNA, you ordered it, it's arrived, you put it into your organism. Now comes the actual bit where you need to see if it works. And like that's always hard. This is something that has been immensely tricky, and we've spent many hours in the lab getting good at, but it's more art than science a lot of the time. Alrighty. Okay, so for analysis, there's sort of as many techniques as you can dream of. You know, these are sort of the ones that I've got here are some of the most common, but there's like 100 variations of each of these that you can do, and there's so many more that I just don't have the time to go through. But these are the ones that are the most basic and you'll do the most. So if you're looking at DNA and you want to see stuff about your DNA, be it is it the right size, or did you PCR a product of the correct size, or whatever, then you're going to be doing a gel electrophoresis. So this is basically you take a block of a special type of gel, and I've got a video on doing this if you want to see it. But you take a block of gel, it's got these little holes in it. You can see the wells at the top of the gel here. There are these little slits at the top. You put the DNA solution in there, and then you use electricity to drag the DNA down through the gel, and it'll separate based on size. So from that, you can actually go, okay, yes, my piece of DNA is the right size, or my PCR reaction worked because it made a product of the right size, or whatever the case may be. If you're looking at proteins, then you're looking at an SDS-Page gel, which again, it's electrophoresis, so it's a special type of gel. It's a different kind of gel, but it's gel. The difference being that for DNA, the gel is horizontal, so it runs like flat, whereas proteins, it's vertical, so it runs down. But at the end of the day, it's the same basic concept. You put a solution in one of the wells and you use electricity to drag it through. Again, it separates it by size, although with SDS-Page, you can tweak the gel itself to separate based on things like the charge on the protein, because, you know, different proteins will have different charge, so you can separate them that way. A slight modification of SDS-Page is what's called Western blot. So this is when you're trying to find something more specific, or you aren't necessarily extracting the protein. So let's say that you want to know if a specific protein is in a particular tissue sample. You're not it's not like you're growing that protein at enormous scale and extracting it into a pure state. Like, you just want to know that it's there. So you would run an SDS-Page gel, so it spreads the proteins out, and then you have to very carefully, and this is the weird bit, like move the proteins out of the gel onto a piece of nitrocellulose membrane, which you can then soak in antibodies that target the particular protein that you're actually looking for. And then there's a whole bunch of ways that you basically make it so the antibody generates a colorful thing when you expose it to a chemical. So this way you end up, instead of lots of lines, you end up with just one line of your target. And so you can see, you know, is your target there? How much is it? Like, how present is it? And that's sort of what that's for. Then you have what's called HPLC, or chromatography. This is high performance liquid chromatography. And this is basically, it's a column of resin or silica gel or whatever. And you put a chemical mixture in the top, and you basically pull it through the gel. And again, it will separate based on its charge or whatever type of, like, you choose how it separates based on the type of resin you stick in there. And then there's a machine that's basically measuring the liquid that's coming out. And you basically take little samples of the liquid that comes out, and it's, so you can sort of separate out a chemical or a protein that's exactly the size or characteristics that you want, and get rid of everything else. But you can also see, like, how pure is your sample. If you get lots of peaks, there's lots of things in your sample, whereas if you get one peak, it's nice and clean. Another one that is basically a combination of, like, Western blot and, well, it's sort of like a Western blot. but instead of on a gel, you just do it in a tube. It's called an ELISA. So basically the way this works is you have a 96 well plate, or however many well plate you want to do this in, and you will stick your sample to the bottom of the plate, and then you will treat the samples with different combinations of antibodies that will stick to your thing, and then you include another reagent that will generate a color if the antibody sticks. So based on the amount of color it generates, it can tell you how much of your thing is present. So that's an ELISA. At some point I'll do I'll probably do videos on how to do all this stuff. I've been thinking of the sort of the best context to do this, but like ELISA is a very powerful technique, and it was one of the ways that we've like proved that we were making silk originally. Although I obviously can't show any of that, but it's neat because it's very visual. Like you look at it and go, oh, it's blue, it worked. Like I love techniques that you just look at it and go, oh yeah, it's working. That's nice. Instead of having to go, I mean, I don't know, stick it in a machine. Does the machine say it's working? Alrighty. But yeah, so like I say, this is a very, very small spattering of techniques. But to be able to do genetic engineering, you kind of need to know how to do all of them and have the equipment to do all of them. I don't have an HPLC. It's one of the ones that I want desperately, but they're like tens of thousands of dollars, so I just can't justify that purchase yet. And more to the point, I don't have a place to put it. But the rest of them are much cheaper and easier to do generally. Western blot and ELISA can get expensive because antibodies are expensive, but you know, a PCR gel is dirt cheap. You know, an SDS page can be very cheap. The other ones get expensive because of antibodies. All right, so once you've done your analysis, what happens? Well, you do it again and again and again and again and again until your thing works, or it doesn't, or you break down mentally. Dealer's choice. I say this not to be flippant, but because like this legitimately happens. Sometimes you'll have an idea and you'll spend months and months and months and months doing it and lots of money trying to make it work, and it just doesn't. Like for whatever reason, it just will not work. At that point, you need to basically go back to square one and go, was my requirement, like was my idea like fundamentally bad? Like did I assume something incorrect? Or is there something that I didn't understand? Or is there something that I need to learn? Or is there a technique that I need to try that I haven't tried yet? On and on and on and on. And it's really where the experience comes in is knowing when to pull that ripcord of this is not working, and then either pivot or give up. Personally, I am very stubborn, which is why I make a good biologist, because I don't give up easily. So, you know, I'm willing to just keep hammering at a thing until I make it work. But it's a hard thing to do, honestly. Like there is a reason that the running joke is you're not a real biologist until you've cried into a 𝐻𝐵. Like this will break you mentally if you're not careful. Like you have to be aware that you've got to like know when to take breaks. You need to know like when to put a project down and come back to it, because there'll be times where just nothing is working. Everything's broken. You don't know why. Like you're questioning your own abilities. You're questioning, you know, just every aspect. And sometimes you got to just put it down and walk away and do something else for a little bit. And then as you're doing something else, you're like, oh my God, I figured it out. And then you know, can go back and try your new idea and maybe try something else. Or maybe you chose the wrong organism. Like maybe you're trying to make a chemical and you just chose a bad organism. Like maybe the thing just can't be made in yeast. Maybe it has to be made in an E. coli, or maybe it can't be made in either of them. Maybe it needs to be made in a plant. Like whatever the case may be, you might have just made a bad assumption, and you know, you got to just sort of you know, go from there. And you know, I would generally recommend sticking with it longer than you think, but not too long. Like there's definitely a point where you're like, this is not working. So for example, I found out the hard way that yeast suck. I hate yeast. They're horrible. They're horrible to work with. They're expensive. It's a pain in the ass. It's just not worth it. It's not worth it. I like E. coli. E. coli is fantastic because it's easy. But you know, I spent months and months trying to design everything to work in yeast before I realized, you know what? Screw this. E. coli are just better. So I switched to E. coli and everything works better. So, you know, it's one of those things that you're just going to have to do it. And like sometimes the only way to learn is to just try. And sometimes it fails, and just be ready for it, and like know when to stick with it and know when it's time to pivot. But, you know, again, you just got to remember biology is really hard. Like, you know, the physicists like to complain about how hard their job is. They have nothing on biology. Like with physics, if it doesn't work, it's a machine. You just tweak the machine. Like, then your engineer did a bad job. Like, or you know, you didn't understand the thing. But like with biology, sometimes your thing just dies. And you're like, why are you dead? What did you do? I didn't do anything. I don't know. Like, or like it mutates. And you're like, why did you change one letter? Why did you delete all of my code and save only the antibiotic resistance gene? That's really frustrating. So yeah, just remember it's hard. Like, remember it's hard going into it, and like, don't give yourself too much grief. Know it's hard. Know there's a huge learning curve, and there's a lot to learn. There's the reason I waited this long to make this series is because I wanted to make sure I knew enough so that I don't accidentally guide people into pitfalls that they're going to get stuck in. And I think I'm at a point now where I can do this, which is why we're doing this stream. But I mean, that is the reality of it. It's really hard, and you just got to stick with it sometimes. All right, so bonus step number 8. Let's say it all works. Now what? Right? Like this is the bit where, like, you know, you got to really embrace your Uncle Iroh and go, and then what? Like, you know, and this could be really simple, right? Like it could be just publish a paper, make a video, make a blog, whatever it is, put the information out into the world. That's totally valid. It's a totally usable, like reasonable output of your thing. But then the other option is commercialize it. Like, again, you could have made a thing that's very valuable. Like those glowing plants are very pretty. They're also worth a lot of money. Like there's a lot of people who are going to want to buy that. So yeah, it's nice to make it for a paper, but it's also nice to get paid. So, you know, this is where it can get very expensive. And then you're dealing with a world of patents and biotech and all of that nightmare. And it's hard. I'm not gonna sugarcoat it. It's really, really hard. But it can be worth it. Like, it can be worth it. Biotech is the future. Like, if you come up with a thing and it's worth a lot of money, there's a pretty reasonable argument to run with it. Like, you know, doing a startup and like raising that initial capital is really, 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 really hard. And I know a lot of people who struggle with it. But if you know your product is good and you know your thing works well, sometimes it's worth it. And, you know, but these are also the thing. It's like, do you want to be an academic or do you want to be in industry? And sometimes the answer is both. And sometimes the answer is neither. And sometimes you just want to make a video about it, or you want to make a blog post or post on Twitter, whatever. But this is it's a very personal choice, and it's also going to depend what you're doing and in the environment that you're doing it. Because if you're in an academic lab, a lot of the time they'll patent whatever it is that they do, not because they want to commercialize it, but because they know that somebody who wants to commercialize it will buy that patent off of you. So while I don't like patents, they are the water we swim in, and it's the way that people get paid. And, you know, that sucks, and I hate it, but it is a way to force, you know, commercial companies to publish their thing. And if you find an old patent that's older than 20 years, you get to use it for free because it's expired, and then everybody's fair game. This is, of course, a problem because people find new things and then hold on to it for 20 years, and that sucks. But we're in this very interesting point in history where one of the reasons why biotech is becoming so popular and becoming such big industry is because a lot of the patents on the original biotech are expiring, which means lots of people are able to pick them up and actually run with them now in a way that they just literally couldn't before. You know, I don't like bureaucracy. I don't particularly like the way that business is done. I don't like capitalism. However, it is the water we swim in, and there's a lot of money to be made, and there's a lot of good that can be done in the world if you just, you know, grit your teeth and do the thing. And this is kind of the reality that I've come to of biotech is the future. There's a lot of money to be made. There's a lot of potential, and you can do a lot of good. Like when you think about the people who spent their entire academic careers trying to make plants that produce higher yields, They're the reason why there are billions more humans on Earth. There are people who are happy and healthy and well-fed today in ways that our ancestors never could have dreamt of because of this technology. And sometimes it's worth swallowing your pride and a little bit of your ideals so that you can get the thing out to people. And a lot of the times, the only way you can do that is patents, and it sucks, but it is the way that it is. But yeah, so those are kind of your options. Now, in terms of actually getting started on all of this, because like, again, this is literally just an overview of all of the things that you're going to need to know and do to be able to do this. I wanted to make sure that I put together a list of actually good resources to help you get started. And now, first off are books, because I have people ask me literally constantly, what books do you suggest? What books do you suggest? What books do you suggest to do genetic engineering? And so I bought a ton of them, and I read them all, and these are the ones that I think are the most useful and are they cover the most ground. Now, like I say, because biology and genetic engineering is a game of knowledge, like the more you know, the better. You really do need to know how every part of a cell works, or at the very least, you need to have a general understanding. You don't need to memorize every step of the Krebs cycle, and any freaking teacher or professor or whatever that tries to get you to do that can respectfully shove it up their ass, because it's just not a useful, well, it's not a reasonable use of your memory stores. The reason it's important to understand the Krebs cycle, though, is that it's the core of biology. So you need to know these things. You need to know how a cell works. You need to know how cell membranes work. You need to know how membrane-bound proteins work. How do pumps work? How do like ion pumps? How do efflux pumps work? How does DNA work? How is DNA copied? How does RNA work? How are proteins made? You need to know these things. So the textbook that I really liked for this was Essential Cell Biology. I got the 5th edition. The most recent edition is, they're all the same. Like, biology is getting constantly updated, but I mean, when you're talking about the basics are all still basically the same. I particularly like the illustrations, and I think the examples and stuff that they give were pretty reasonable. So if you have no background in biology, that's probably the book I would start with. Then if you want to actually get dip a toe into the waters of genetic engineering and synthetic biology, the middle 2 books are fantastic. So Biobuilder, I think I've recommended many times on the channel before, but I actually bought a copy and read the whole thing, and it's fantastic. Like, it does a really, really, really great job of explaining how genetic engineering is done. It actually includes protocols, which is cool, but it's a really great primer. Synthetic biology, a primer, also very good. It covers things that are it covers a few different topics. It's a little bit older, or it feels a little bit older anyway. But the things that it includes are really, really interesting pieces of information, especially in the context of genetic circuitry and having genes that interact with each other and with themselves, and trying to do things that are more complicated and really pushing the bounds of synthetic biology. It really gives a good taste of that. I have a lot of genetic engineering experience, and there were things in there that I had never heard of before that I then immediately became fascinated and slightly obsessed with. Things like the repressilator, which is this really, really cool bit of genetic logic that we will go through at some point, and I desperately want to have made. But it basically is a system of 3 proteins that repress each other in a cycle. So only one of the proteins is active at any time, and it's like a built-in genetic timer. And people have actually made it, and they once you have this cycle going, you can have it drive expression of whatever other proteins you want. So I found a paper where they built a repressilator that would have 3 different fluorescent proteins turn on depending on which spot in the cycle it is. So as the little E. coli colonies grow, you can actually see rings in the colonies of the different colors, because it's like they're turning on as the bacteria gets older and as the colony grows. So there's a lot of really good stuff in there. And then the last one is Concepts of Genetics. I don't actually think this is as useful of a book compared to the other ones. It's much more complicated. It gets really into the weeds of the really, really nitty gritty of what you can do with DNA, but it also spends a lot of time on like Punnett squares and population genetics and all the stuff I find very boring. It's good to know, but I don't find it particularly interesting. For somebody who's getting started, though, it's probably worth the read. Just not to memorize it, but just to have more familiarity with the sorts of things that can be done with biology so that you can dream bigger and think of bigger, more complicated projects and things that you want to do. In terms of YouTube channels, I mean, these are the ones I recommend the most for people getting started. I mean, Crash Course barely needs an explanation. It's the thing that's gotten me through school. It's gotten many people through school, particularly their biology, chemistry, and organic chemistry courses are phenomenal and literally teach you basically everything that you would get out of like a first year university class. They're fantastic. I highly recommend them. They're really, really great for beginners. Again, highly recommend. iBiology is a really neat one because it's not the basics. It is not the basics by any stretch. This is getting into like deep into the weeds of basically the craziest things I've seen anybody do with biology. They're basically professors giving lectures on the different things that they've done. And like, if you think what like the shitty biohackers are doing is interesting, you have no idea what academics can do. Like, these people have access to essentially infinite resources and infinite time, and they make very good use of it. Like, these are people who are studying so there's a whole series on like super resolution imagery and microscopy, and you know, there's series on neuroscience and the development of the brain, and just anything you can think of, they've got a lecture on it. They're not necessarily the cleanest of lectures. Like, it is still ultimately some professor just talking at you with a lot of usually very cool pictures. And sometimes they will skim over the basics. So like, they sort of expect you to know more, but that's kind of okay. Sometimes when you're trying to learn about something new, just dive into the deep end and just sort of like let the information wash over you, and then watch it again, and then try and understand it better, and then maybe do some Googling, maybe read some papers, and then watch it again so that you try, like, all the pieces start to like fit together in your brain. And so I think it's just really valuable to see sort of the width and breadth of what can be done. So again, it's a fantastic channel. I highly recommend it. But in terms of extra resources, I've put some more links in the description, but these are sort of the ones that I recommend the most, and I think will get you the furthest for the least amount of effort and input. Like I said, next time we're going to be going through building plasmids and really actually getting into, okay, now how do you do the coding? Because in the Who's Genes streams that I've done before is, you know, I tended to glaze over a lot of stuff. Like, I'm just working the way that I'm working. Like, I'm doing the research and finding the things and putting the pieces together. But there's things that I do that just people don't understand why I would do it that way or why it's done that way. So I want to really do it properly. I want to go through the process of, okay, this is how you build a plasmid. These are the pieces. These are why they're important. This is how you this is how they go together. And this is how you put it together. Because building DNA is like most of what you end up doing in a genetic engineering lab. Like, most of the cycle for me in my like day-to-day is you come up with a design for a thing, you order the DNA, and then you spend like, unless you're having it made entirely, which is usually more expensive and slower. A lot of the time you have to order individual pieces and then assemble them. And the way that you do that is going to dictate how you design the code. So yeah, a lot of that is where you have to like keep all that kind of stuff in mind. So we're going to go into the weeds. We're going to like really go into detail next time on that one. But yeah, that's the end of my little presentation here. So with that, I'm going to open it up to questions. I hope you guys have all enjoyed this little overview. I'm not sure when we're going to do the next stream. It's kind of dependent on when I can find enough free time to like sit down and, you know, have the time to like do the stream. But they're gonna generally be on weekends, and I really want to try and do one a month, because I think that's a pace I can keep up with, even though My work during the day is very, very busy and very intense. I think I can still do one stream a month and hopefully some shorts and some videos and stuff intermingled in. But yeah, that's the presentation. So I'm gonna set this to just camera. Now you get to see my beautiful face and my ridiculous hair. Yeah, like I say, I'm gonna open it up to questions. I've had our moderator keeping an eye on the chat the whole time, and so they should be sending me a list of all of your questions that they saved throughout this whole thing. And but I'll also just I'm now looking at the chat. So if anybody wants to ask a question, now's your time. Ask away. Okay, so I've got the list. So first off, can I post the slideshow afterwards? Yes, I will post it on the GitHub. I have a GitHub. I don't know if it's linked below. If it isn't, I'll link it after the thing. And I'll post the slideshows so that way people can follow along and go back through it afterwards. Okay, next. Do you have any experience using 𝐷𝐸𝐵 plasmids? No, so I can't give it any advice. Sorry. What else we got? Is genetic engineering and protein engineering synonymous? I wouldn't say that they're synonymous, but they do go hand in hand. So a lot of the time, the way that you're doing protein engineering is via genetic engineering. So it's a cycle. So you'll make a protein, you'll code for it, make the DNA, express it, test it, and then go, okay, I need to make changes. And then you go back to the code, change the code, make the new protein, rinse and repeat. What else we got? How's the bioluminescent Christmas tree coming along? I mean, that was sort of a one-off video. I'm not going to really be coming back to that, but it was fun. So what's the difference between glowfish and neon tetra? Nothing. They're the same, as far as I know. As far as I know, neon tetra is either a brand or it's a subset of glowfish, and so they're the same thing basically. Can you make yeast produce things like vitamin? Yes. I mean, that's what the beta carotene yeast was. It's producing vitamin A, or at least it's producing a chemical that gets turned into vitamin A when you eat it. But I mean, any other vitamin is totally doable. The efficiency is always the question, but to just make a vitamin in a yeast is not necessarily difficult. You could do vitamin D, you could do vitamin C even, although that one's a little weird because vitamin C is acidic, so you can screw with the actual growth of the cell, and that gets a little screwy. But yeah, no, totally doable. Did the milk polypeptides have correct glycosylation? I have no idea. I didn't get that far into the testing. The first step was just seeing, did the yeast that makes the milk protein, does it make the protein? And the answer is yes, but the level was really, really low. But I want to revisit that at some point because I think there's a way to do it better than it was. What else we got? Did I get a major immune response from the lactose thing? And the answer is, I think so. I was pretty sick a couple days after, but the problem is I was also traveling, so I'm not sure if it was an effect of my immune system reacting to the new expression of proteins, or if it was I just don't travel well, or I maybe got sick while I was in the plane or something. So it's hard to say. I suspect baby, but I can't say for sure. What else we got? Have I read Project Hail Mary? No, I don't know what that is. Is there a risk that a protein in one organism is a prion for another? I would say generally no. Prions are natural protein. Like the only things that are prions, like they're proteins that exist in your biology already, and the only difference between a prion being prionable and not is I mean if it's misfolded, but also like the main thing that causes prion disease in humans, right? is a difference of a single amino acid is the difference between it being capable of folding wrong. So people who have a mutation in that gene literally can't get prions. Like if you've got the right version of it, you are completely resistant to prions. You can like get stuck full of it and nothing will happen. So the chances of like a random protein being prion-like is very low, I would say. I wouldn't say it's impossible. I would say it's very, very low. Okay, what else we got? What are the limits of genetic engineering? I honestly don't know. I don't think anybody knows. Because every single day, genetic engineering gets easier and we get new techniques that let us do new, harder things. Making things that are not physically possible, probably not. It's limited by physics, really. Like it's physics, chemistry, and the functions of biology. So like you're not going to be able to make an organism that can like withstand 1,000 degrees. That's not going to happen. Like biology is incompatible with that. But you can go pretty far. Like you can go pretty far. Like making Captain America, probably doable. Probably doable. Maybe not to the it's not a 1-to-1, but like I could make your bones stronger. I could make you probably bullet resistant. Like, we could make you immune to all kinds of different stuff. Like, there's a lot you can do with that. But, there are limits, of course, because, keep in mind, we're not, like, the universe has hard rules, and you gotta stick to them. What else we got? Can you genetically engineer macroscopic organisms, like hot pink fluorescent apples? Yeah, of course. No, that's super easy. I say super easy. It's easy in the sense of like it's doable. It's not easy in the sense of like the actual act of it is necessarily easy. But yeah, it's doable. Pink apples? Yeah, that's fine. Like, pink apples is easy. Like, it's one gene. Whereas, if you want to make like, I don't know, dinosaurs, that's really hard. That's a lot of genes. And also we don't know enough. So that's really tricky. Can Canadians genetically engineer tourniquets? Tourniquets? I recognize that word. I just don't remember. What's a tourniquet? Oh, corals. Oh, okay. They're like marine invertebrates. Maybe. I don't know. I don't know the laws. I would assume so, but I would need to check. I don't know. How do you manage your documentation during an extended research period? Okay, this is actually something that like every single day is a new challenge, because the more you do genetic engineering and the deeper into the weeds you get with it, the better your documentation has to be. So this means extensive lab notes, lots of labeling, like coming up with organization systems to keep track of all of your DNA and all of your samples and all of your stuff, figuring out an organization system. One of the things I'm really lucky for is that one of the people I work with is fantastic at organizing stuff. And, she's spends a lot of time just making sure that everything is neat and tidy and we know where to find everything and all this kind of stuff. But it's a lot of work. A lot of the work that I spend, like a lot of the work during the day is just keeping things organized and making sure that everything is noted down. And because if you don't write it down, you'll forget, and then you're going to end up having to redo the experiment, and nobody wants to have to do that. So just making sure that you just constantly take very detailed notes is super important. Can I give an ETA on the Spider Silk project? Buddy, if I could tell you when it's done, I'd be a happy man. But I don't have an estimate. So it'll be done when it's done. What else we got? This is actually a really good question. I really like this question. Does paying the publisher to read the paper support peer reviewing? No. Peer review is this weird lie that we all tell each other that doesn't actually do anything. Like, so let me frame this properly. All peer review does is the point of peer review is to see if the conclusions at the end of the paper match the data. So like you can't say, like if your data clearly shows that a thing doesn't work, you can't say that it clearly does work. The problem with this is peer review is just three assholes. Like, it's just 3 people randomly selected, not paid. Like, they're forced to volunteer. They're not paid for their effort. And they will make requests and will force you to make changes to your paper that will distort it out of the actual purpose of the paper. Generally based on whatever that person happens to be interested in, even if it has nothing to do. So peer review does not actually guarantee that the paper is good or that the data is legitimate. People fake data. It's a thing. Like, people leave details out of their method section that makes it impossible to replicate. So when an actual expert reads a paper, there's a very good chance that you go, do these researchers have brainworms? And this is literally a sentence that I've said so many times in the last 3 months, I don't even know what to do with myself. Like, most papers suck, and they've been peer-reviewed. So it's not actually a good judge of whether or not a paper is good. So, and also, again, the peer reviewers are not paid by the publisher. The only person who gets paid is the publisher. Like, researchers have to spend thousands upon thousands of dollars to pay the publisher to publish their paper, and then the publisher makes thousands upon thousands of dollars from all of the people reading the paper. The only people who get paid are the publisher. Fuck them. Excuse my French. So yeah, no. So, what supports peer reviewing? Abuse of academics is mostly it, which is depressing, but it is the reality. Yeah, right. Like the Alzheimer's drug. Like, we just found out that like 15 years of Alzheimer's research was basically bunk because the original people lied. But it was a peer-reviewed paper. Like, there's this huge problem of, like, we peer review papers that are not good. And like, when you actually dig it, like, eventually somebody only catches that the original people lied when they try and replicate a paper, but publishers refuse to publish replication papers. So, like, they'll only publish things that are novel, which is useless and not how science is supposed to work. But again, that's because publishing was designed by a capitalist fuckhead and has essentially ruined science. So, or not ruined science. Like, science still continues, but in spite of publishing, not because of it. Anyway, so that's that rant. Okay, what else we got? Any recommendations on an affordable -80? Yeah, the shuttle freezers. So there's a company called Shuttle, and they make these like cooler-sized -80s. I'm not going to sugarcoat this. It's still thousands of dollars to buy one, but I mean, I managed to get one for I think $2,000, which sounds like a lot, but for a -80 that like works and is portable, is crazy, and it's quiet, and it's wonderful. I love it. If you're going to buy a -80, that's the one to go with if you're just starting out. You can usually find them on eBay for pretty cheap. Like, they don't show up all the time, but sometimes you'll find one and it's like $2,000 and it's worth every penny. Anyway, so that's that one. Why is there so little content about genetic engineering in the lab? Because most people don't make videos. I do. But I mean, I'm busy. So like, I'm busy getting good at genetic engineering so that when I make videos about it, they're actually good. But it's not like, there will be more videos personally. Like, personally, I will be making more videos on genetic engineering. I'm just not in a place right now where I can. I've been like, part of the issue is I had a lot of projects fail. It's biology. Things fail all the time. So I've had a lot of projects fail, which means I can't make a good video about them. But I've been finding that and I also have a lot of projects that are too small to make a normal video, which is why I've been starting to make shorts. So for those of you who saw the first short that I made, it was about a bacteria that produces an enzyme that lets you make a banana smell, amongst others. That video will be a good video when it's done, but it needs more work. But I can make a short about it. So like, that's kind of the trade-off that I've been personally making. Any plans on selling the beta-carotene yeast? No. It's not my code. I'd have to license it from whoever made it. And I don't have a store set up right now. So, nah, sorry. Can I recommend some cool papers about this stuff? There are far too many. Like literally just pick a topic and just they're all fantastic. If you want a really cool paper about genetic engineering, look up the repressilator and all of its variants. It's fascinating. But in terms of a specific paper, I mean, not off the top of my head. Can you make yeast produce estrogen? Probably, but the problem is not the making of the estrogen. The problem is the getting it into a pure enough form for people to use, and that's where the problem is. That step is far more expensive than making the yeast, because if you screw it up, a person dies. So this is why I say, like, if it has to do with medical stuff, it's always harder, because the regulations and the requirements, so you don't accidentally kill somebody, are very stringent. So it's a hard thing to do, not because the genetic engineering is hard, but because the chemistry and physics of it is hard, and regulations are hard. Any recommendations for learning more about plant genetic engineering? Okay, so if you want to learn about plant genetic engineering, the fellow that I mentioned earlier in the stream, Sebastian 𝐎𝐫𝐢𝐠𝐢𝐳, at a tiny green cell on Instagram, Twitter, blah blah blah. He also has his lab is called 𝐎𝐵𝐢𝐺 Labs. Check him out. He has some phenomenal guides on plant tissue culture and genetic engineering. He's really great about open sourcing everything he does. He's just a great dude. I would recommend checking out his stuff. He's got really fantastic stuff. What purification methods do you typically, like protein purification methods, do you typically use? That depends. That really depends. It depends what you're doing. Sometimes you get lucky and your protein is weirdly soluble in something, more so than anything else, like vinegar. Or you don't get lucky, and then you have to use His columns and His tags and all this kind of stuff. Or you need an HPLC, which is the only way that you can do it without like a tag. But it's hard. I mean, it's a lot of the time it's various chromatography methods. How much money did it cost to put your lab together? A lot. It's a lot. I can't give you the number, but it was a lot. Okay. That's everything off of the list that my my fantastic mod has collected. So now I'm gonna just go through the comments, and I figure we'll give it another maybe 10 minutes and then call it for the day. I really hope that you guys have enjoyed. This has been a lot of fun. I'm really glad that I'm back to doing streaming, and I hope to be doing it more in the future. Okay. So here I'll put. Oh wait, no, cancel. Okay. The name of the guy is 𝐵𝐢𝐺 Labs, or tiny green cell. There you go. It's in the live chat now. So if you're looking for him, you can find it. I'll put a link to Sebastian's stuff in the description after the stream is done. Somebody. So this is a comment I get a lot, and I want to address it, which is people are like, I'd be interested to see the failures. No, you wouldn't. There's nothing to see. The problem, the whole reason I don't make a video if a thing fails is because there's nothing interesting to see. Like the nature of YouTube, fundamentally, is people will only click on the most interesting thing. Nobody wants to sit through half an hour of here's a bunch of goo that doesn't work properly. Like, people want to see the things that work, and there's just not enough. Like, it's too much effort to make a video about a thing that doesn't work, as opposed to like nobody's gonna watch it, and it's a lot of effort, and it doesn't pay, and it doesn't, like, it's not useful information. Like, the reason I will only make a video about stuff that actually functions is because that means I've solved all of the problems, and it's now a thing you can follow along with. Whereas if it's not functioning, it might just not work, and I don't know why. So I can't make an interesting video about going, oh, I'm shit out of ideas. Like, it's not interesting. So that's why I don't do it. It has to be actually interesting and actually purposeful and finished. It's got to be finished. Like, I will not let myself publish a thing that's not finished. And that's how I've always viewed it, and it's how I'm going to keep viewing it, honestly. Okay, what else we got? I'll take a couple more questions, then I'm going to wrap it up for the day. Again, the reason I don't make videos about failed experiments is because I don't necessarily know why they failed. I just might have a pile of broken garbage that doesn't work, and I go, I don't know why. Why are papers so hard to read? I don't personally find them hard to read, but I've also been reading them for a very long time. The problem with papers is that they are written in a very specific format and tone, and they're presented in a very specific way that if you're not accustomed to it, it can be hard to parse. But it's one of those things you just got to do. Like, the only way you're going to get good at reading papers is just read a lot of papers. I wish I had a better answer than that, but I mean, that's just the way it is. Okay, what else we got? Have you done more work on the opals? Yes, I have, actually. I have worked on the opals. And then the fucking cylinder got knocked over. that was bad. But I mean, it's neither here nor there. I got to make another batch of opal. And I've been working on a modified protocol that will actually end in large, high quality opal. It's just the problem is the settling process takes literally months. So it takes forever to do the experiment. So it's taking ages and ages, because you got to like do the reaction. That's a whole day of like setting it up and running the thing. And then you've got to settle the particles out over however many weeks or months. And then you got to not bump it or it gets ruined. And then you got to get the liquid out and then dry it. And it's a whole thing. So I'm working on it. It's probably just going to take me a long time to get it good, because it's just hard. It's just hard. It's just the way it is. It's just hard. I also want to just take a second to like thank everybody who's donated. I don't have the donation bar up today because I don't have a specific goal in mind, but I do appreciate the donations. It helps keep the channel going, and it also gives me the motivation to like keep doing the streams and keep doing the stuff, because I know you appreciate it, and I just anyway, thank you. Very, very much appreciate everybody who pitched in today. Okay. The typical educational route to a career in yeah, okay. So here's the problem. I don't know the typical route to a career in genetic engineering because I didn't take it. I would say the most typical route is to do an undergrad in some flavor of biology, or biochemistry or molecular biology, and then doing a master's or PhD that's more focused on like synthetic biology. The problem is there aren't a lot of synthetic biology programs at the undergrad level, so they're hard to find. But if you can find one, I mean, they're great. Probably. I wouldn't know. I've never taken one. But some of my friends who are now doing their PhDs, like that was the route that they took. But I mean, it's also not the only route. I know people who are now in industry and who are leading biotech companies who they might have done their undergrad in bio, but then they just had an idea and ran with it and cooked something up themselves. And now they've got a whole big company that they're running. So it's everybody's journey to get into genetic engineering and to be doing it professionally is going to be a little bit different. And so I can't tell you how I did it because I didn't do it in the regular way. I did it in the dumbest possible way that cannot be replicated. Nor would I suggest replicating it. It was not a fun process. It was a lot of like strife and hardship, and it was not fun. But yeah, just like but for most people, just getting started, do an undergrad. Like, academia has its flaws, but like it's really good at teaching you the basics. If you can afford it, or if you're in a sane country that provides it for free, as it should, then take an undergrad, do it. Like, life is short. Learn some stuff. It's fun. That's it. Okay, what else we got? I'm gonna take one or two more. What jobs can you get with a biotech degree? I don't know. Working in a lab somewhere. You can either work in industry or you can work in academia. Those are your choices. Find a company that's doing a cool biotech thing and either work as a technician or as a lead scientist or as a research scientist or whatever. Work for the pharmaceutical industry, work for the agri-tech industry. Like, there's a lot of things, anything you can dream of, and then some. Like all these companies that I mentioned, like Mufree or what are they called now? Perfect Day, or any of these other, like Light Bio. Like, find a company that's doing something cool, apply and work there. Like that's how those are your job options. Do something cool or work in an academic lab and become a professor eventually. Okay. What else? What's your opinion on community biohacking spaces? They're hit and miss. They're hit and miss. I have a lot of problems with biohackers. My patience for them is evaporated. It's basically just people trying to do genetic engineering poorly most of the time, and that's really frustrating. I don't want to say that, I don't want to use too broad of a brush there. There's a lot of people doing a lot of very cool stuff, but the problem is that a lot of people are sloppy, just so sloppy, and they don't think shit through, and they don't plan it out. And there's like, they never ask the and then what question. It's always like a lot of excitement, and then nothing works, or they find out how expensive it is, and then it fails and it goes nowhere. And it's frustrating. However, if you can find a community biospace, there's a couple of good ones, and like they do some cool stuff. A lot of the time it's like, mostly growing mushrooms, like edible, like gourmet. A lot of the time it's working with 𝐸𝐲𝐈, but sometimes it's like, there's the open insulin people are based out of a community space, I believe. And they're doing really great stuff. So it's, it's hit and miss. Yeah, we're biopunk because we do things haphazardly. Right. Like that's the thing, right? Like it's just doing things poorly. And it's really frustrating. Like, just do it properly or don't do it. Like, there's a phrase that, anything worth doing is worth doing badly, which is to say it's better to do it badly than to not do it. But with bio, it's very expensive. So doing something badly is just wasting money a lot of the time. And like, then it encourages people to quit. Like, one of the reasons I hate the 𝐎𝐷𝖆 and all of their stuff so much is that their stupid kits are usually so low quality that like they don't work, and then people go, oh, this is too hard, and then they give up. Or they've just, taken stuff from people without a license or whatever else. And it's really frustrating. Like they like brand themselves as, we are amateur gods. And it's like, no, you're just some schmuck. Like it's frustrating. I'm of the opinion if something's worth doing, do it properly or don't do it. And it generally works out. If you take the time and you think it through and you plan out your thing and you have like a goal and you have a purpose, it's generally better than if you're just flailing. And there's so much flailing in the biohacking community. And it's really frustrating because I know a lot of people who do really good stuff and they get drowned out by idiots who are doing really bad stuff. And it's hard. It's hard. But that's not to say it's impossible. There are people doing good stuff. Sebastian being one of them. There's a few more, but that's kind of the thing. Okay, what else we got? I'm gonna do probably one or two more, and then I gotta call it because my voice is killing me from talking for 2 hours. Right, like this. Okay, yeah. So somebody's like, I like the biospace here in Cambridge. Great bunch, cool ideas, and sharing the cost. Like that's where biohacking spaces or like DIY biospaces shine. It's where like you've got a great community and you've got a great group of people coming together to do a thing or a small group of things, and they share the cost and they share the work and they work together to do something really awesome. And when COVID started, there was a huge push of these like DIY bio people trying to come up with ways to help. And a lot of that, like a couple of them actually got spun out into real companies doing actual stuff. And a lot of it was just flailing. It was like, let's 3D print masks. It's like, you can't 3D print a mask. You just can't. You can't 3D print an N95 filter. It's just not possible. So, there's a lot of good. You got to just be careful. And the only way you're going to know, honestly, is to just go to your local DIY biospace, if such a thing exists, and meet the people and talk with them and see what they're doing and see what their space looks like and see if it's mostly cobbled together garbage or if they actually have like some decent tools. And, it's okay if you're using cobbled together garbage as long as you're not also trying to like save the world. Like if your autoclave is a pressure cooker and your you know, your sterile hood is an inverted box with some holes cut in it, that's fine. You can do lots of cool stuff with that, but you should probably stick with like growing mushrooms rather than trying to do anything complicated, because the complicated stuff's expensive and it's just not going to work if you don't spend the time and the money on the thing properly. So, you know, it's a mixed bag. Is it hard to engineer algae? I'm going to tentatively say yes. I haven't tried it, so I don't know for sure. I know I have a buddy who does it. It's more difficult than E. coli and it's more difficult than yeast, but it is doable. Like there's a small number of papers on genetically modified algae, and I really think that that's probably the future of biotech because it solves so many of the biotech problems. There just isn't a lot of literature on how to do it, so it's a lot more striking out in the dark and sort of figuring it out as you go. And look, and yeah, so somebody's like, growing mushrooms is fun. Growing mushrooms is great. Mushrooms are delicious. And like you can grow gourmet mushrooms very easily with very few tools. And it's a really great way of getting started doing genetic engineering, or like at least getting used to the microbiology aspects of it. And then at some point, like you'll get bored of mushrooms and go, you know, I really wish I had an E. coli that made a this, or I really wish I had a yeast that did this. Then you start planning it out and figuring out, okay, what's this going to cost? How am I going to do this? What are the places where I'm probably going to get stuck? These are the questions you have to be asking as you're trying to do this thing. But yeah. Okay. I want one more, and then I'm calling it. Are bacteriophages the future for medicine? Yeah. As antibiotics become harder and harder to find new ones, phage therapy is probably going to become very, very potent, because there's a lot of diseases now that are getting harder and harder to treat because they're becoming more and more antibiotic resistant. Like tuberculosis is getting much more difficult to treat. Like there was this distinct dip where tuberculosis was very treatable, and then it became very difficult to treat again. And so, you know, phage therapy is really interesting because it's very difficult for a bacteria to be both resistant to a phage and an antibiotic. So it's almost always a trade-off. So most of the time you can use the phages to weaken the bacteria and then use the antibiotics to like finish them off. So you'll go from something that's resistant to every antibiotic to something that suddenly is not, and one of the antibiotics starts working again, and now you can actually treat the disease, and it goes from something that's lethal to treatable. So yeah, I think that is a pretty solid thing, and there's a lot of really interesting genetic engineering that goes into making better phages that are more lytic and are more targeted and are more broadly applicable than their original host organism. Yeah, so that's phages. Okay, anything else really fun? Otherwise, I'm going to call it for the day. Okay, there's a bunch of questions here. So there's somebody keeps asking about the quote brain drug video. That was the drug in question was cerebrolysin. I took it down because I mean it just wasn't as professional as I wanted, and it in the space that is YouTube these days, it just doesn't fly. So I took it down because it's like better take the video down than risk getting my channel taken down. Was the math there. Okay, I'm gonna call it there. This has been a lot of fun. I hope you guys have enjoyed. If you have recommendations, or if you have things that you want to learn about in the next streams, leave me a comment. Like I read every comment. Just leave me a comment, and I'll try and work it into the next streams. But yeah, with all that being said, I hope you guys have enjoyed. This has been a lot of fun. I'll see you guys on the next one. There'll be some new shorts soon, maybe a video soon, but we'll see. And definitely more streams. So yeah, have a wonderful rest of the day wherever you happen to be, or night if it's night there. And yeah, I hope you all have a great time. Have a good night. Bye.